Okay, so welcome to the week 6 lecture series of the live NPTEL live sessions of the course Cell Culture Technologies. And so today we will be discussing about the neuronal cell culture. So neuronal cell culture is the kind of like it's a little difficult because neurons of all the cells like the skin cells, the cells of the intestine or cells of the epithelium, the cells of the lungs or heart. So those cells they can divide and they can, those cells can show cell division and growth. But neuronal cells, neurons especially, they are very difficult to they are very difficult to grow and divide after a certain point of time, so they stop their cell division after a certain point of time. So neuronal cell culture is difficult in the sense that whenever this kind of cells are isolated or this kind of cells are grown in culture dish, these are this we have to be very careful so that we don't lose such cells, because otherwise we have to again kill another animal, sacrifice another animal to culture those cells. Mostly these neuronal cells, they are grown in primary cell culture, and they do not form cell lines because they do not have regeneration capacity, so they have very poor regeneration capacity or very poor cell growth, as a result of which they are not very suitable for converting into secondary cell line or secondary or the continuous cell line. So these are mostly this kind of cells are isolated from the animal brains and animal spinal cord tissue, and they are cultured in dishes, and for some certain experiments are performed using those cells, and then they are discarded after a certain point of time. So they are not grown continuously. So this kind of cells are basically very difficult to culture, and so we have to be very careful when we are dealing with the neuronal cell culture, because there are like problems like this where if this gets contaminated, or if it is and also because they are very like resistant to cell division, so it is also very much possible that like when we are isolating cells from the brain, so when we are isolating cells from the brain tissue, it is not only composed of neuronal cells, but also it is composed of other cells, accessory cells like glial cells or the astrocytes. So if such cells are present, so it might be possible that the actual neuronal cells, they are not able to, they will not be able to grow, and this kind of contaminating cells will outgrow the number of the neuronal cells. So it is very we have to be very careful so that this kind of accessory cells do not outgrow the neuronal population, and we need to be like cautious enough so that we do not end up contaminating the cell culture with other kind of microbes and viruses. So an ideal system for culturing the neuronal cells should be similar to physiological condition, which should be true for any kind of cells, not just neuronal cells. Any kind of cell culture, if we are performing, we have to mimic the conditions that is similar to physiological condition. And the best idea to mimic the physiological condition is to grow the cells in a 3D area where there are cells plated in layers. So layer 1 and layer 2 and layer 3, that way. So when cells are plated in this kind of layers, so the cells show interactions with each other, not only in the 2D plane but also in the 3D plane. So culturing the cells in such 3D culture resembles more of that of a physiological conditions, and so it is like if we are testing certain drugs, or if we want to mimic the exact physiological condition, it is always suggested that the cells need to be grown in 3D. Otherwise, if other signaling cascades need to be studied, or if any kind of apart from signaling, if any kind of interaction, any kind of effect to the cells, whether the cells are dying or whether the cells are alive, those kind of effects can be studied in 2D environment as well. So so neuronal cell culture. So here in this course, sir has only taught about neuronal cell culture, but some characteristics are common for all kinds of cell culture, irrespective of whether they are neurons or whether they are other parts of the other parts or other organs cells. Some criteria like myelination, or the criteria of conducting neuronal impulses. So some specific characteristics are need to be maintained when we are culturing neurons. Apart from that, all other characteristics for cell growth or cell culturing conditions remain the same. For example, these cells are also grown in 37-degree and 5% carbon dioxide, similar to all other cells. So the general idea remains the same, that how the cells need to be grown, but there will be certain differences with the medium that the neuronal cells are using. I had talked about the medium that it uses some in some previous class. I'll again discuss it in this class. So we need to mimic the conditions of the neuronal cell culture, which should be similar to the physiological conditions, so that it will allow cell-cell interaction, just not the cells we are working on. Like it should not just be the neuronal cells. It also should allow the interaction between the neuronal cells and the glial cells, and neuronal cells and the astrocytes. So interaction between neuronal and glial and neuronal and astrocytes should be present. This is astrocytes. So this is glial cells and this is astrocyte cells. So basically all these accessory neuronal cells, the glial cells and astrocytes, they are associated with providing nutrition to the neuronal cells, and that is also very important for the healthy growth of the neuronal cells. So once we are isolating the mixture of cells from the brain or from the dorsal root ganglion or from the spinal cord, and once we are dissociating all those clumped cells and we are culturing it in the dish, we should make sure that in order to mimic the proper physiological condition, because under physiological conditions it is not like there is just a single cell that is growing, but in any kind of organ there are different multiple tissues or multiple cells that makes up like the multiple cells interacting with a single cell. So these interactions are also very much important for the activity of the particular cell. So, mimicking the physiological condition, and exactly when we are culturing the cells from the in vivo system to in vitro system, we have to keep in mind all these characteristics, so that it does not deviate from the physiological conditions. Like when in brain, when the cells are growing, they are in different conditions, and when we are culturing it in vitro condition, it should not be a very drastic change in environment. Then it will lead to misleading results that we do not want. So always we have to make sure that our culture that we are isolating from the brain, it should resemble the physiological environment, and it should show similar kinds of responses, how similar kind of responses and similar kind of interactions, how it would have shown in vivo condition. So there are different kinds of neuron which is present in the central nervous system and in the peripheral nervous system. So the central nervous system is mainly composed of the brain and spinal cord. So it consists of brain and spinal cord, and whereas the peripheral nervous system is of all the other nerves which connect from the spinal cord to the different area of the body. So the neurons which are present in the central nervous system and peripheral nervous system, their structure is different, in the term that peripheral nervous system we need a longer axons, because we need to transmit the impulses. For example, here you can see the peripheral nervous system. We need to in peripheral nervous system, we need to have a longer axons. This axon should be longer as compared to the central nervous system. Central nervous system is limited within a small area, but the peripheral nervous system, nerve impulses are carried from one part of the body to a very distant part. So this axon should be very much longer, and in order to insulate the axons, we have squam cells. So these squam cells go and bind, and they basically encircle like this. So in this particular axon, they will the squam cells will reach over here, and then they will encircle this part of this axon, and it will cause insulation, so that when these nerve impulses are being transmitted over longer distance, the nearby cells which are present near to this near to these neurons, these cells should not get affected by the nerve impulses that are being transmitted. So squam cells, in case of peripheral nervous system, they kind of encircle these axons and provide insulation, and this kind of insulation that is present in the central nervous system is called peripheral nervous system. It is called the myelination or myelin sheath. So this is known as the myelin sheath. Myelin sheath. So this kind of insulation is called the myelin sheath, and we have squam cells providing this myelin sheath in case of peripheral nervous system, whereas in case of central nervous system, this is again divided into two parts: central nervous system and enteronervous system. So these are again two different subgroups where of central nervous system itself from the brain. From the brain, when the nerves, they are the nerves that is transmitting the impulses from the brain, they don't need to transmit it to very distant areas like that of spinal cord. Because the region inside the brain is very still in under the reach. So in the brain, when the nerve is transmitted, the axon length is not as long as that of the peripheral nervous system, and also the protection against the different cells, against the electrical impulses that are transmitted through these axons, are provided not by the squam cells but by another kind of cells that is known as oligodendrocytes. So these oligodendrocytes, they form this kind of processes which extend from this kind of cells. So these cells, they form kind of processes, and these processes are extend to the axons, and they again encircle the axons and form myelin sheath. But this myelin sheath is lesser lesser in amount as compared to that of the peripheral nervous system. As you can see over here also, the amount of the amount of the myelin sheath formation is not as much as that of the peripheral nervous system, because the impulses are transmitted within a very smaller region, and so it's a little bit of insulation is fine. And so brain and spinal cord. So spinal cord is in the peripheral nervous system, and brain is in the central nervous system. So the that the transmission of neuronal impulses between brain and spinal cord through happens through different neurons which have different characteristics, and these characteristics need to be kept in mind when we are culturing neurons either from the CNS or from the PNS. The first step for achieving myelination in the peripheral nervous system in vitro conditions from an embryo are written here. So basically, the embryonic rat. So the this kind of isolating the neuronal cells from adult has still not been optimized, and there are certain challenges with the adult. So this kind of neuronal isolation or peripheral neuronal isolation happens mostly from the embryonic rat. In the embryonic age, like about 14 days old embryo. And because of that, in this embryonic rat, which is about 14 days old, the dorsal root new ganglion neurons, or DRGs, basically these DRGs these DRGs are isolated from an embryonic rat. So this embryonic rat, they are dissecting the brain from this embryonic rat and getting the dorsal root ganglion here somewhere, and this from the dorsal root ganglion they will so from so they are isolating the dorsal root ganglionic neurons from the spinal cord region. So when they are dissecting this particular embryonic rat, they will take the spinal cord region from this rat, and this rat, the spinal cord region, they will dissect, and then they will put it in like some trypsinized condition, because they will not get it as separate isolated cells. So from this from the spinal cord region, they will first isolate the spinal cord, and then they will cleave it using trypsin or other kind of isolating other digestive enzymes. And once these are cleaved, and once we get cells which are isolated instead of clumps, then these cells, we can, from these cells, through their morphological differences or through their density based differences, we can isolate the dorsal root ganglionic neurons. So we have to, when we are using the trypsin to dissociate the tissue, so this enzymatic dissociation using trypsin should be stopped within 10 to 15 minutes using a trypsin inhibitor, because trypsin, it's a peptide, it cleaves peptide bonds, and the peptide bonds not only is present between the two cells, so basically the cell clump here, so trypsin will help to cleave this cell clump. But if the trypsin is used for longer period of time, this kind of because it's a peptide it's a peptidase, and it cleaves peptide bonds. So if the trypsin is kept in the medium for longer period of time, then the peptide bonds inside the cells as well, inside the cell, will also be cleaved. So the cells also will start getting killed. So we have to use trypsin inhibitor, and this reaction, like the trypsinization, should be stopped within 10 to 15 minutes. It should not surpass the 10 to 15 minute timeline, because if there is more trypsinization, then the peptide bonds inside the cells will also get cleaved, instead of just the peptidic bonds between two cells. So once the enzymatic dissociation using trypsin needs to be stopped at 10 to 15 minutes time. And so after the dissociation is done, then we have to plate all the DRG cells in the dish, isolating the DRG cells, and then identifying the DRG cells properly and then put it in the dish. And when they are grown in dish, some of the cells will have extended processes. Some of the cells will have extended processes, and they will grow. They will grow longer axons. So these axons then will show the characteristics of neuronal cells. So from a mixture of different cells, like from a mixture of glial cells, from a mixture of astrocytes. So neuronal cells need to be identified that way, and also we need to do further some analysis to confirm that the neuronal cells that we are growing is ideally the DRG cells, or ideally the particular neuronal cells and not any contaminated cells, through immunochemical studies as well as through some morphology based analysis. Although morphology based analysis might not be a proper proof, there should be some immunochemical assays that should be performed to confirm that that particular cell that we have isolated is of a particular cell type. So every cell type on its surface have a particular indicator or particular protein that is that indicates that the cell type is the same cell type as we want, the one which we want. So those receptors are targeted using certain antibodies which can detect those receptors. And so basically, if a particular cell has, so for example, T T helper cells. So T helper cells have CD4 receptors on their surface, and so antibodies against CD4 receptors can identify T helper cells because they are having so much of CD4 receptors. So if we are culturing the T helper cells in a dish and we want to identify whether that cells are whether those are actually T helper cells or not, then we can confirm by using antibodies against the CD4 receptors and growing and then checking by fluorescence. Like we have to label the antibody. So the antibody which is identifying CD4 receptor should be like, suppose is the primary antibody, and then the secondary antibody should be labeled with should be tagged with some kind of color. Suppose red color, this is the secondary antibody, and this secondary antibody should have a colored group, red color or green color. And so we can, if there is fluorescence observed, that means the culture that we are growing is having CD4 plus cells. So apart from just culturing the neuronal cells, we also need that the neuronal cells should get their Revant, you have any question? Revant, can you turn off the mic? Okay, so when we are growing the neuronal cells, once we are sure enough that those are neuronal cells and they are having axons, and so they have certain like some kind of morphological similarities to the neurons, that is not just enough. We have to mimic the physiological condition. So we have to also add the Schwann cells in case of peripheral nervous system, because in case of peripheral nervous system, as we saw, the Schwann cells are associated with myelination. So these Schwann cells we need to add in the media, in the cultured media containing the neuronal cells. So these Schwann cells are isolated from rat or rat at postnatal state of P1 or P4, and this will cause myelination and release myelin basic protein. So this is myelin basic protein. So as in the class also, sir had explained that when these Schwann cells reach the site of axon, they will start getting dividing. They will start dividing myelin basic protein. So when this protein, I will just write it. So when the Schwann cells reach the site of the axon, they will have the these Schwann cells will start dividing, and then they will form a layer on the surface of the axons, and they will start secreting certain chemicals which is known as the myelin basic protein or MBP. So this will allow these cells to lose their identity. So now instead of these individual cells will no longer be individual, and they will start behaving like a common a common tissue like thing, and will start coating the surface of the axonal cells with it will start coating the axonal cells with this kind of protein and structure and a lot of Schwann cells. So this will form a insulating band or insulating protein which will be coated on the surface of axon, and which will be known as myelin sheath. So this is the myelin sheath. Myelin sheath. And so there are different there will be different Schwann cells migrating towards this axon, and they will form myelin sheath on the surface of this axons. So it is myelin sheath is not continuous, because the Schwann cells, whenever they are reaching here, and they will start dividing, and then they will form this network of MBP protein and myelin sheath formation. So there will be some gaps between the each myelin sheath, and these gaps are known as nodes of Ranvier, and these gaps are very small, and there is no covering, but that does not matter, because most of the neuronal axon is covered, and so the different so it is not going to cause any kind of harm to the nearby cells. So we are going. So now has these nodes of Ranvier and this myelin sheath. They have certain certain distinguished characteristics, like the myelin sheath secretes myelin basic protein or MBP, and the nodes of Ranvier, they are having a high concentration of sodium channels. So they have a high amount of sodium channels. It could be the high amount of sodium channels because they need to conduct nerve impulses, and nerve impulses conduction requires the depolarization of the membrane, and that happens due to sodium and potassium imbalance. So this kind of characteristic features of nodes of Ranvier or the myelin sheath is important in recognizing that the myelination has been done properly or not, by using antibody against the MBP protein, and by using antibody against sodium channels. So if the antibody recognizes the MBP protein properly, and the sodium channels properly, so MBP protein should recognize the myelin sheath, and then the sodium channels should be recognized by another antibody, again MBP, again another antibody. So there should be, if we are tagging it with two different colors of antibodies, say red and green, so this should be red and green. So the pattern, if it is followed like this, that means the myelination has happened. So another way to isolate the neuronal cells or the peripheral neuronal cells from the peripheral nervous system is like first. So here, as we had first isolated the DRGs, and then we had put the Schwann cells, but the next step, we can form the bed of myelinated cells or Schwann cells, and on top of that we can add the neuronal cells that needs to be cultured. So but we have to be careful that when we are adding Schwann cells before. Schwann cells, they are not they have regenerative capacity, and so they might outgrow the number of the neuronal cells that we want to grow. So if that happens, then our main purpose for growing the DRGs or the neuronal cells is not met, because the Schwann cells are heavy in more in number, and their characteristics will be present in the and their characteristics basically will be present in the entire dish mostly, and the characteristics of neuronal cells will be masked. So we have to restrict the division of Schwann cells by using antimitotic compounds that will restrict Schwann cell division. So again, this compound should be used in such concentration, and only those chemicals which can inhibit the growth of only Schwann cells, and should not affect the neuronal cells. And apart from that, so first criteria is to put the neuronal cells and then put the Schwann cells. Next way of growing this kind of cells is first put the Schwann cell and then put the neuronal cell, and next way is to grow both the Schwann cells and neuronal cells together or co-culture. So all the ways are possible. It depends upon which way you are comfortable in working with, and which way the lab has optimized their cell culture growth conditions. But how to make sure that the myelination is happened? So again, we have to make sure that the three characteristics have been achieved. One is the conduction velocity. So conduction velocity depends upon how fast the neuronal cells are conducting the neuronal impulse. So this velocity is measured, and it has to be compared between the myelinated cells and non-myelinated cells. So they will show differences in conduction velocity. So if this conduction velocity is proven to be correct, and if it is if myelinated cells and non-myelinated cells shows differences, then we can be sure that myelination has happened. Also, we have to use antibody against MBP and sodium channels, which will help us recognize the myelin sheath and the nodes of Ranvier. And that will help us believe that the myelination has properly taken place, and the neuronal cell culture is also resembling the physiological condition. Just take some. So next, this far we were talking about how we can achieve myelination in the peripheral nervous system. Now we'll be talking about how we can achieve myelination in the central nervous system. So the central nervous system, as the central nervous system has difference from the peripheral nervous system neurons, because the neurons in the central nervous system is is actually the neurons from the central nervous system has oligodendrocytes for causing the myelination instead of the Schwann cells in case of peripheral nervous system. Just 1 second. Yeah. So although the main idea remains the same, that we have to dissect out the brain from the rat, and or the spinal cord from the rat, and then culture it. So in case of central nervous system, we have to dissect out the brain. In case of peripheral nervous system, we have to dissect out the spinal cord. And once the brain is dissected from the rat, the neuronal cells can be cultured from the different parts of the brain, the hippocampus, cortex, brain stem, or cerebellum, depending upon what is the requirement and what characteristic feature we want to study. So as the neuronal cells, so suppose we want to culture the cells from the hippocampus. So now in this hippocampus region, there is another collection of different neurons, pyramidal neurons, oligodendrocytes, microglia, astrocytes. So all these neurons that are different types of neuronal cells, sorry, not neuronal cells, there are different types of cells that are present in the hippocampus region, of which the pyramidal neurons are the actual neuronal cell that can conduct the neuronal impulses. Rest are all accessory parts, and they help in either providing nutrition or providing insulation or providing the different support to the growth of the neuronal to the actual neuronal cells. So each of these cell types, when we are separating brain, when we are taking out the brain from the rat, and we want to culture those cells, so each of these so when we are culturing the cells, there will be a combination of different cells, pyramidal cells, oligodendrocytes, microglia, and astrocytes. So all of these cells are present in the same mixture, and we want to separate it so that we can get each of these cells as a separate entity, and we want to grow it in separate dishes. So in order to do that, in order to grow each of the cells in separate dishes, we have to perform density gradient centrifugation. So in density gradient centrifugation, we can use either sucrose, or another was Optifex or something that sir had discussed in the class. So OptiPrep. So OptiPrep is also similar kind of material. It is just I think better suited for neuronal cells. But generally also sucrose gradient can also be used. So basically what happens is different concentration of sucrose depending upon what gradient, like basically how so we need to have some idea about what kind of like cell mass will be there. So we have to know that the cells that we are working on, or the cells or the mixture of cells that we are working on, what should be its approximate molecular weight, or what should be its density, and what should be like how which should be bigger and which should be smaller. So now that it is optimized, but the earlier days it was not optimized, so people had to do different permutation combination to isolate different types of cells, and then grow them, and then after growing them check the different marker levels and check the morphology of the cells, and finally they came to a particular conclusion that using this centrifugation density, or using this speed, we can achieve the isolation of particular types of cells. So here this is just an example. So different gradient of sucrose, say 5%, 35%, and 40% sucrose is been used, and it is when it is undergoing centrifugation, so samples with different densities will be isolated along with the density at which the sucrose was plated. So here there will be a different set of sample, here there will be a different set of sample, and here there will be a different set of samples depending upon how much heavy the samples we have. So here you can see that the centrifugal force is applying towards the down, like towards this side. So centrifugal force is applying here, and there is a density gradient. So the centrifuge tube is filled with a density gradient. More heavier gradient is will settle down here, and lighter molecules will settle down on the top. So this density is maintained throughout the column or throughout the test tube, and this is then the sample is then applied on the top of this centrifuge, and then the sample and the centrifugal column having different densities of substances. These are the density is towards the like downwards, so it increasing density is from top to bottom. So once the sample is applied, and then the mixture is centrifuged, and we can get different distinct columns, and we have to very carefully isolate each of these columns and place it in separate dishes, and we have to culture them in separate dishes. So we will get cells of different characteristics and morphology, depending upon in which layer it is getting deposited. So depending upon whether the cells that we are using is high molecular weight or bigger cells, if bigger cells will get pelleted before, so this will be bigger cells, and then this will be smaller cell as compared to this, and this will be very small cell. So depending upon the molecular weight of the cells, and depending upon the molecular weight, and depending upon the size of the cell, it is getting density of the cell, it is getting precipitated. And so using this kind of gradient centrifugation, we can isolate a particular type of cell type from a mixture of cells. So after density gradient centrifugation, what we are supposed to do? So after the density gradient centrifugation, very carefully, we have to pull the cells which is present in different layer, and it is placed in individual dishes, and they are allowed to grow. So each of these cells will now grow into a independent colony and independent morphology, and they will they will grow, and then they will show not they will not be a mixture of cells, but an individual colony of cells, and so they will show their individual characteristics, and we can use those cells which we want in our which we want to continue our experiment with. So cells are then further characterized, so based on immunocytochemically, or cell morphology, or the conduction velocity, as we were discussing even in last time. So similar to peripheral nervous system, how the neurons were characterized, similar way only we have to characterize the central nervous system neurons as well, but there will be little difference, because central nervous system we also have oligodendrocytes which are associated with myelin sheath, so we can use antibodies against protein which is expressed in oligodendrocytes. So if it is properly attaching to the axons, then we will see fluorescence. So we have to know that against what protein we need to design antibodies, and against what region we need to design antibodies, so that our characteristics of the cells that we are growing, it resembles to the actual characteristic of what cells we want to grow. And so this has to be this should not be like kind of just a general knowledge. It should be very specific to the cell type that we are working with, so that there is no confusion regarding what cells we are growing. It is often it is very like it oftenly it can happen that there are cells from different parts, but they look very same, their morphology is very much same. So if their morphology is same, it is very difficult to distinguish that what cell type it is. So from the first itself, when we are working with cell culture, we have to we have to keep in mind that we should not contaminate one cell type with another cell type, and we should ideally maintain each of the cell type separately, and only in cases where we want to co-culture them, like in this case where we want to co-culture the Schwann cells as well as the neuronal cells, or we want to co-culture the oligodendrocytes and the motor neurons. Oligodendrocytes, or the neurons from the central nervous system. So during those times, we have to use like we have to again refer to methods which scientists have already standardized, and not waste our time into standardizing our own methods. So it is very important that we go through the papers which the scientists have already standardized and optimized over a period of time. So next is the embryonic motor neurons culturing. So as for till now we were seeing about all the embryonic peripheral neurons, the neurons from the central nervous system. So here now we need to culture the embryonic motor neurons. How we can do that is like the mouse embryo, about 12.5 days. So those mouse embryo is taken, and then the spinal cord is removed from the mouse. Then again the enzymatic treatment and trituration of the spinal cord, which will cause the breaking of the spinal cord, and the presence of the tissues clump will be dissociated, and there we will get single cell suspension of cells. And this single cell suspension of cells, we have to plate it on lectin coated culture dish. So lectin is again the ECM matrix, which is favorable for growth of motor neurons. So once this lectin coated culture dishes are placed and plated with the single cell suspension, which is obtained from the lumbar spinal cord, we will get dissociated spinal cord cells. So SC stands for spinal cord. So SC is actually spinal cord. And MN is motor neurons. So MN stands for motor neurons. So once the dissociated spinal cord cells are present, this motor neurons will get attached to this plate, and then we have to select those cells which are which has motor neurons, because lectin will select only the motor neuron cells. And then we have to wash it with HBSs. HBSs is a buffer which has all the components which is similar to the physiological buffering condition. Then we will have only the motor neurons which are attached from the mixture of spinal cord cells. Only the motor neurons are attached, and then we have to detach the cell with depolarization solution. So depolarization solution will depolarize those neurons and then detach those neurons from the coated dishes. And then we will have another suspension of cells which will have only motor neurons, and these motor neurons are then finally counted on cell counter, hematocytometer and hemocytometer, and then they are finally plated on the culture vesicles to culture vessels to grow them. So this is the enriched motor neuron culture that is grown. So either the culture is grown either in the cover slips or in glass or in dishes. So depending upon like what cells we want to culture on, so we can also ideally modify the ECM, and then we can select it. So another way of selecting particular cells of particular cell type is immunoseparation. So immunoseparation again uses the concept that there is a particular set of antibodies that is plated on the surface of the dishes. So this antibody, suppose, recognizes only a particular receptor which is present on the surface of neuronal cells. So only neuronal cells are going to attach on the surface of this antibody coated plate. and so only this cells, only the neuronal cells will be growing on this surface. So then we can select only the neuronal cells, while all other cells will be removed using washing. So if you are washing it many times, then only the cells which are very strongly adhered to the surface, they will only remain, while the after washing the cells which are very weakly it was attached, they will be removed. So we can get a pure culture of cells of a particular cell type. So this is known as immunoseparation. So again, we will just go through the hippocampal neuron growth requirement. I had also discussed it in last class, some classes before. So neurobasal growth medium is used as a medium ideal growth condition for hippocampal neurons. So it's supplemented with also antibiotic, antimycotic for prevention against contamination. Consists of glutamax, which is a dipeptide of glutamine and alanine, and poly-D lysine coated plates are present, which is suitable for hippocampal growth neurons. So here lectin plate is suitable for motor neurons, but for hippocampal neurons, it is mostly the poly-D lysine coated. Media composition and culture conditions must be optimized so that a mixture of hippocampal and glial cells are present. The glial cells, due to its capacity to divide, should not overpopulate the non-dividing hippocampal neurons, but we should have to maintain the proper interaction between the glial cells and the neuronal cells, and we have to optimize it, that how much amount of media we have to use, how many different kinds of anti-mitotic inhibitors we have to you have to treat the cells with. So it is all about the knowledge of what cells we want, how much we want, and how much like what kind of experiments we are going to perform with it. So that determines a lot of the time that what characteristics or what conditions the cells need to be grown. So that is all, and we will now move on to solving some questions. If you have any other doubts, you can ask me.
Ma'am, can you explain conduction velocity?
Okay, so conduction velocity, it is basically a measure of how much current or how much electrical impulses are passed through the particular axons of a neuron. So suppose if the neuron is bare and it is not having any myelin sheath on top, then the velocity that it will pass through the impulse with, it can be, so there are methods by which this velocity can be measured. So suppose there is any kind of electric, so just normal condition, if there is any electrical wire with no kind of insulation or no grounding, then if somehow that electric wire is at fault, and you can, if you touch it, it will be like the velocity or the current impulse you can measure, that will be like directly you are measuring it, so it will be very high. But if it is myelinated or on the surface of the on the surface of this axons, if there is coating, or if there is a like a protective shield, so now if we are measuring the velocity or the rate at which the current is passed, that will not be directly from this membrane region where actually the current is passing. So current is passing through this region, So here if it is bare, then the measurement from this region will give a very high conduction velocity, as in like because it is directly in touch with the axons. But if it is having myelination on the surface, then because it is insulated now, it is not the measurement will not directly be from the part of the neurons which is conducting the electrical impulse. So this will show a lesser amount of speed of velocity, like the speed or the current impulse transmission. So basically conduction velocity, as the name implies, is the rate at which the impulse is conducted through the axons or through the neurons. So the speed at which the impulse is being carried. So it will be difficult to, so if it is a myelinated neurons, we should have a different conduction velocity. If we are measuring the conduction velocity of those neurons, it should not be similar to those neurons which are not myelinated. And so it's an important characteristic feature to identify myelinated and non-myelinated neurons from.
In the cell culture, how, ma'am, they will measure it?
So in the cell culture, so have you heard of machines like patch clamp, or those like where they can in real time measure current? So there are certain machines. So in what happens is, so basically these cells, the cells on their surface only, there is some kind of pipettes. Okay, so these pipettes can, so inside the pipette there will be some buffer solution which is equal to the buffer solution of the bodily fluid, and like similar to the buffer solution that has to be present in the cell. So what it will do, this pipette solution, what it does is this sucks the cell like this, and it creates a small kind of hole, not like hole, but it forms a continuous membrane. Like suppose the pipette is here in this cell, it is touching the cell, and then if you suck the pipette from this part, then the cell here, it will form a continuous membrane with the pipette. Like the cell, instead of like once it is touched here, so here the cell membrane is intact and the pipette is just touching it. Once it is once the that is called a shield formation or giga shield formation. So once we pull this pipette from here due to the suction force, this will now form a continuous membrane, a continuous structure with this pipette. So now what happens is, here there are electrodes placed basically. So one electrode will be placed over here and one electrode in the cell, and so from this from here they form some kind of the connection, like how the positive electrode, negative electrode, there is some kind of physics that allows the that has a concept of physics in it, that will allow the forming the circuit basically. And this circuit you can use, like when you are putting these electrodes over here, we can in real time measure the current of that particular cell. So in neuronal cells also, similarly, in the neuronal cells that we are culturing from primary cells, or if myelinated, whether it is myelinated or not, then those cells also we have to use some techniques like this, which can measure the current conducting through those cells, like the how much current is flowing through the cells, and so that can be measured in real time using this certain kind of sophisticated instruments like patch clamp.
Thank you, ma'am.
Does axon diameter affect the conduction velocity?
It should actually affect the conduction velocity, because according to the if the diameter is thinner, then it should be much more faster conduction as compared to thicker axon. Like I don't know, like some formula is like the electrical wires had some formula. I don't remember it clearly, but I remember that there was some formula which depends upon the diameter, like not diameter, the radius actually. So if the axon thickness is like different, then it can actually differ. But in this case, if you are saying in this case, because we are isolating it from the same like from the same organ, suppose if you are isolating it from the brain, then the neuronal all the neuron, and from the same brain, I mean from the same organism's brain, so it is not going to have much effect on the like the axon thickness might not be different. Like I can, like, maybe you might be thinking that the axon thickness can also show differences in conduction velocity, and how do we know that whether it is myelinated or not myelinated, or whether the axon thickness is actually causing difference in the conduction velocity. But here in this case, as we are isolating it from the same animal and from the same organ basically, so the axon thickness is not going to matter over here. It might be different for different like different species or different animals, but from the same animal, it is very less likely that that can happen, that axon thickness is affecting the conduction velocity like that. But to answer your question, axon thickness can conduct the can change or can affect the conduction velocity.
Thank you, ma'am.
Welcome. Okay, so now let us move on to answering the questions. Okay, so the first question is this. So some book for exam preparation. See, I have personally only gone through certain handbooks of different brands, like this Thermo Fisher, or the Sigma, Sigma, Sigma or Mark, MERCK. So those have worked for me, but as in like cell culture book, I am not like like I have not gone through any cell culture book, but also there are certain papers. I mean, there are certain scientific papers that have described like neuronal cell culture, or the basics of cell culture, basics of cell culture. Mostly the handbooks from different companies, they have a good amount of information regarding the basics, but as the, like, according to the classes as it is progressing, it is not just basic cell culture, it also has like all the primary cell culture and all the techniques for isolating those cells. So those, I think it will be better if you like study by topic, like search on Google, and you can get certain good papers, scientific papers, and I think that is better way to gain knowledge. As in, like, you don't have to read the entire paper, but you can read the abstract and some figures. There will be some graphical it's like very nice I find graphical abstracts or representations that are present in those papers, which answers the cell culture cell culture technologies very nicely. So you can go through those. Okay, so only one person has given the answer. What about others? What should be the answer of this question? What is facts, and how, like, what is the basic of separation of facts? So there is a huge confusion, it seems. So FACS, basically it has three components. It's the uh, fluidics, optics, and electronics. Please ignore my handwriting. Fluidics, optics, and electronics. So these are the three components. Sorry. So these are the three components of FACS, and so it depends upon. So fluidics is mainly, basically, it has a very thin capillary through which all these cells are passed. So this is a very thin capillary, and the cells are passed in such a way that at a time only a single cell will be passed through light. So the fluidics part is when the capillary is having different cells. So this cells is a mixture of cells basically. And this capillary is so thin that at a particular point of time only one cell passes from through the light. So this is a light source, optical source, and that is forms the optics part of FACS. And this optical source, light source, passes light through the cells. So once the light passes through the cells, based on its scattering nature of the, so we know that light has electromagnetic properties. It can scatter and it can even deviate. So it has both particle nature as well as wave nature. So depending upon how the light is scattering, we can get different populations of cells. Suppose the cell is like there are two types of scatter: forward scatter and side scatter. So forward scatter is FSC, and side scatter is known as SSC. So based on the forward scatter means how the light is scattering from this region, and side scatter means how the light is scattering from the inner part of the cells. So side scatter shows how much particles are present inside the cell, how much basically how much granules are there in the cell. So this is like, it will give an idea of granules. And forward scatter gives an idea of what is the size of the cell. So bigger the size, bigger will be the forward scatter. But the main concept of how the light, how it is separated, is based on the electromagnetic nature of the light and based on the scattering phenomena of the light. So because based on those, because of how the light is scattered and how the cells are cells can have granulated structure as well as non-granulated structure, and the different sizes of cells will show different scattering type of the light. Therefore actually it is not it is not a chemical based separation, it's actually a electromagnetic based separation, because here the cells are being separated on the characteristics of how it is scattering the light. So, and light shows some electromagnetic properties. So it is like the answer is option B. What is the full form of FACS? FACS is fluorescence activated cell sorter. Okay, you can write it, fluorescence activated cell sorter. Okay, I will write also, because I don't have a pen. It is, my handwriting is not very good. fluorescence activated cell sorter. Okay. So yeah, so let's go to next question. So the question is motor neuron. Okay, MOTOR, motor neuron. Okay, so a lot of you have a little confusion, I think. So here, so basically, this is the spinal cord, and the back portion of the spinal cord is the dorsal horn, and the front portion is the ventral horn. Horn in the sense that there is a gray matter here, and here it is dorsal horn, and here it is ventral horn. So so it is like the sensory neurons. So the sensory neurons are actually taking the sensations from the sensory organs, and then putting and then they are giving the information to the dorsal side, like from here. And from the ventral side, the motor neurons are allowing the responses through the motor activity. So it is not option A. Mostly of you have written option A. Only one has written option C. So actually the motor neuron, it is present in the ventral horn. The sensory neurons, they are conducting the impulses through the dorsal horn. So the option is correct, that is ventral horn. Okay, so it is fluorescence activated cell sorting only, as far as I know. Pavi, it is, I think, fluorescence activated cell sorter or cell sorter or sorting. I mean, cell sorting is the technique and sorter is the machine. So, yeah. Okay, so now let's go to next question. Okay, very good. Most of you have given the correct answer. So, immunopanning is a process which is employed in cell culture to identify and separate different cell populations based on immunomarkers. So, it is a technique of immunoseparation. So, it helps immunoseparation. So basically, it allows the plating or panning of antibody specific to a particular receptor on the surface of the dishes, and here the mixture of cell is poured. Only those cells which are having antibody specific receptor, so this is the antibody, and only those cells which has the receptor to this antibody, I will just write here antigen, but no, I will not write. So receptor to this antibody, those cells will bind here. Rest all the cells, they are washed and removed. So cells having a receptor which is specific to this antibody, so antibody receptor interaction happens, and then the other cells which do not have that antibody are washed off. So we can get populations of different cell types using immune markers. So we can use so a mixture of cells. Suppose we have 5 cells in this mixture, and each cell has a different antibody, different receptor on its surface or a different marker. So we can plate 4 different or 5 different dishes with different markers. So 5 different dishes are having antibody A1, 2, 3, 4, and 5. So first time we'll put all the mixture in first here. So this is antibody 1, and so the receptors which are similar to antibody 1 will bind. The cells having the receptors will bind over here. Rest of the cells are washed and removed. So the next mixture of cells from here, like the cells which are attached, the rest of the cells which are not attached, they are moved from here to here. Then again, whatever cells are attached is here, and again the residual media is again put over here. So it continues till the time all these cells are cells having the specific receptors are attached to their respective antibodies. So the correct option is option B, and so it's not option D. It's not just to isolate fluorescent cells. So yes, immunohistochemistry, definitely the antibodies are used for isolating fluorescent cells and detecting fluorescent cells. But in terms of cell culture, it is not the prime goal. It is one of the major goals here. Immunopanning does not mean just isolating fluorescent cells, but detecting cells which are having a particular marker, so that from a mixture of cells we can isolate only a particular cell type. So option B is the correct answer. Next question. So what is the answer? Only one has given the answer, I guess. What happened, rest of you? So it's not collagen. If it was collagenase, it could be an option, but collagen is not the correct answer, because collagen is the material which helps in cell to cell adhesion. So collagenase is okay. What's wrong in getting clumped? So the problem is that when the cells are clumped, it might be the individual characteristics of the cell is not, you will not be able to say whether that is the individual characteristic of the cell or whether it is the characteristic of the entire clump whenever we are doing the experiment. For like as in like in our case, when we are doing an experiment of imaging, or if we are doing a microscopic experiment and if we are getting a clumped cells, it is very difficult to select cells first of all, because if they are clumped in such a way that suppose they are clumped here, so it's very difficult to select whether this is an entire cell or whether this is an entire cell, or whether whether these three are different cells. So it is very difficult to identify the individual characteristic of the cell. So when we are doing culturing the cells, we want to see what is the characteristic of this individual cells, not as of a tissue or as of a clump. So so that is why we don't want the clumping of cells. Okay, so I have got all sorts of answer. A, B, C, D, everything is there. Okay, so yes, actually the answer is DNAs, not vitronectin or collagen or phosphates. So like we have to be careful whether it is like collagen or vitronectin. These are actually chemicals which help in cell to cell adhesion. Collagenase, if it was instead of collagen, then okay, then we could have considered it, but because it's just collagen, so this option is not right. Vitronectin, it also causes the ECM formation, so it is not associated with removing the clumping. Then phosphates are not, phosphates, they are not present even in the cell adhesion complexes, or they are not they are not associated with any kind of cell clumping, or this is not an enzyme basically. So phosphates, collagen or vitronectin, these are not enzymes. They are just molecules. They are not going to cleave anything. So out of all these four, only one is an enzyme, that is DNase 1, which can cleave the DNAs. And because these are not, like, there are no other suitable options, ideally it should be like collagenase, or it could be trypsin. If the cells are getting clumped, we can use all those. But because out of the four options that is given over here, none of them are enzymes and none of them are associated with cell adhesion. So cell adhesion, removing the cell adhesion. So the correct answer is not A or C or D. It is actually answer is option B. Okay. So
Ma'am, why DNAs? Could you please explain the reason once more?
see, DNAs, it will cleave the DNA, all the the DNA present in the cells. So suppose if there is clumping of the cells, then they are basically degrading the DNA and they are basically killing the cells. So more the number of more, so in this case, so we have to be careful with how much time we are incubating with DNAs, or how much time or what is the concentration of DNAs, because if there is a very high concentration of DNAs, then all the cells will be killed. But if there is a minimal concentration of DNA, that means out of suppose 3 cells which are clumped together, if the 2 cells are killed, then only 1 cell will be remaining, and this 1 cell will be an isolated cell. So here DNA is basically degrading the DNA present in each of the cells. But we have to be careful that how much concentration and how much time we are incubating it, because we don't want all the cells to be killed. And anyway, when we are isolating a primary culture, like it is not always possible that we will get isolated cells, so there will be clumps, and it is okay to sacrifice some cells, because then we will be able to get certain individual cells, and we can carry on the analysis with the individual cells. Always the analysis is performed within individual cells, and because we have to see what is in, and each individual cells will behave very differently from each other. So it is very important that the individual cells characteristics are monitored and studied. So it is fine that if we have less individual cells, then having more cells in clumped condition. So why usually the cells get clumped in the first place? This is the characteristics of cell, because I mean in the body, in vivo system, they are neither in isolated cells, in isolated systems. They will always be present in tissue form, or they will always be together. So their first characteristic, their default characteristics is to get clumped. I mean just before we are culturing it in vitro condition, when they are present inside the body of the individual, they are always present as a tissue form, or they are always present as a group. They are not present as an individual cells. Always there will be some cells that it interacts with. It is not always that the cell is individual cell. It is always they will be either they are present in a tissue where they are interacting with similar cells, or they are present in the circulation where they are interacting with different cells. So in either case they are interacting. Basically their default characteristic is characteristic of all the cells is to interact and to form a group or a community. But in cell culture, in vitro conditions, we are not allowing that community to form. So because we want to study them as an individual cells, which also makes the job more challenging, because that is not an ideal scenario inside the system where the cells are in present in the tissue form and they are always interacting. But nowadays, as the 3D cell culture is coming up, so in 3D cell culture, this characteristic of cell to cell interaction can be maintained without causing the cell to clump, like in 3D in layers. So when the cells are growing in layers, it can be grown in isolated form, but it can still mediate the interaction. So it can still show interaction between each other, but still maintaining the isolated characteristics. So there is a kind of like difficulty to get the isolated cells all the time, because the general tendency is to get clumped. And also if the cells are very stressed, like after trips in treatment and after such isolation from the in vivo condition, they will also be, if they are getting stressed, then also they will start to clump together and they will form one mass. So there can be two reasons, whether either it is due to stress, or or due to the normal tendency of it getting clumped. How the DNAs enter into the cell. So it's an enzyme. So it's an enzyme. It should enter. I don't know. I mean, there should be some transporter or something. I don't know. I have to check. I mean, I don't know. So these are enzymes only anyway, but there should be some transporter or some receptor which can allow the DNAs to enter the cell. So not enter the cell, or either the cell is the cells which are not very healthy, they will have some cracks on their membrane which may allow DNAs to enter, but I am not sure about this. I have to check. DNA does not need to enter the cells. No, it needs actually. The DNA is present inside the nucleus, so the DNAs need to cleave the DNA. So I will check. I will get back to you in the next class, Shweta.
Excuse me, ma'am.
Yes.
Ma'am, when we are culturing single cell suspensions in huge quantities, I mean in the bioreactors where the conditions
Hmm.
are very difficult to control,
Hmm.
then what do we do if the cells are clumping there?
So if the cells are clumping there, then again I think we have to do some enzymatic treatment, and we have to use enzymes which will cause the declumping of the cells. I like there should be certain enzymes which are suitable for using in the in bioreactors, um, or some kind of if we are so basically in bioreactors, if in most of the cases bacterial cells are cultured, so for bacterial cells some other kind of enzymes can be used, which is specific for those prokaryotic system. But if mammalian cell culture, I am not like I'm not sure, like if in big kind of quantities they are used, then also I think we have to use those kind of enzymes only, trypsin or collagenase or whatever. If we are culturing some cells which are having higher amount of cells, particular cell adhesion molecules, so we can test it, like which molecule is causing the cell adhesion, and then they can synthesize an enzyme against that molecule, and then they can put that enzyme in the bioreactor, and I think then the clumps will be reduced to some extent. But if it is still clumping, then maybe they have to reduce the amount of cells that they are putting in the bioreactor. Okay, so now let's go to next question. So extracellular DNA is not causing the clumping. Actually, the clumping is caused due to the cells. I mean, if all the cells come together, that is causing clumping. DNA is just I mean, DNA is it's going to be present. It's not going to cause that kind of difficulty. I mean, it's very small. Like it's a very small amount of DNA. If it is released, also that will be washed off. So it is not going to go and attach to the cells and cause clumping. As far as I know, it is mostly the cells only. The cells are getting clumped together. Okay, I've got one answer. Yes. Yes, so that is the correct answer. So all the four, gravity and sedimentation, magnetophoresis, we can use this technique for isolating red blood cells because red blood cells have hemoglobin, and hemoglobin has an iron center. So we can use the magnetic properties to, isolate red blood cells. Microfiltration depends upon the size of, so we can use filters of different sizes, and then depending upon the filtration size, different cells of different, like larger cells and smaller cells, they can be separated. Optical sorting is again dependent on the antibody based sorting, where each of the cells can be tagged with different antibodies, and then depending upon their fluorescence, we can sort it. So these are all gravity and sedimentation based on density, magnetophoresis based on magnetic properties, microfiltration based on the size, optical sorting based on the specific antibodies on their surface or receptors on their surface. So the correct option is answer C, option C. So correct. Next question. Okay, so the, yeah. Yeah, so the correct option is correct answer A. So this basically it's a separation technique which uses glass micro bubbles. So basically all these micro bubbles are buoyant, or they remain floated in the, in the solution, in the suspension. So these are micro bubbles. I'm labeling it as MB. So these micro bubbles are coated on their surface with antibodies. So the target cells that can bind to this antibody will bind, and so here we'll get some target cells which are bound to this micro bubbles. Because these micro bubbles, they do not settle down, and they are buoyant. That is why it is known as buoyancy activated cell sorting. So these microbubbles which are floating and they have antibody on the surface, they are attached to all these cells, the target cells, whereas the non-target cells, they do not attach to this antibody and they get precipitated over here. So all these non-target cells, these are non-target cells, they will get precipitated here, and all the target cells, these are bound over here. So we will get a nice separation of target cells present on the top portion. So here all the micro bubbles with the target cell will be present over here. So all the target cells are here with the micro bubble attached to the micro bubble, and we will get the non-target cells which will be precipitated down. and so it can be then removed. These are non-target cells, and then this two layers can be removed. So the option is the correct option is answer A. Very good. Now let's go to next question. Okay, so A is not the correct answer. So anything which is dividing from one cell or one clone. Suppose this is one cell only, not a population of cells. Suppose this is one cell. If anything is dividing from this, then it can be called as monoclonal. But here the question is when cells are selected from a culture. So we have a culture, and so there are different types of cells. Okay, means this is an heterogeneous culture, and we are selecting some cells from this culture. So and then we are growing it. Suppose we are selecting these two cells, but the cells which are growing from these two cells are not grown. So this is going to again be an heterogeneous population because it is not from the same parent cell. So this is a different parent cell, this is a different parent cell. So as a result of which we are not going to get a monoclonal population. It is like from different cells we are getting their progeny. So this is not a monoclonal population. And what does monolayer means? You have any question?
Ma'am, what is a sublime?
Sublime means so when we are taking one set of cells to another set, that is known as sublime. Like suppose you are taking from this particular dish to so from here you are selecting certain subset of cells and growing into another culture, another dish, that is sublime. Like from
Yes, ma'am.
one part, yeah, some part of it you are growing into another cell, that is sublime. So that is not monoclonal. Monolayer means if cells are growing in single layer. So if cells are growing in single layer, that is called monolayer. So this is also not correct. Explant means when we are taking any tissue or any like part from an animal and growing it in a dish. So if we are growing any plant tissue or any animal tissue and growing it in a dish, that is known as that plant tissue is basically the explant. suppose that small part of that tissue. So this is also not the correct answer. So yes, so answer is the correct answer is option C, cell strain. So this will give the characteristics. The strain is characteristics of the population, the subset of cells that you have isolated from the main dish, the subset of cell that has particular characteristic, because you must have identified that culture from with certain antibodies or with certain selection. Okay, so if we are giving some antibiotic to these particular cells, and only we see that these two cells are these two cells have survived, all other cells have been dead, so these two cells will now give rise to a progeny of cells that has an antibiotic resistant mechanism. So that particular progeny of cells is known as a strain. So similarly, that is also the strain is also known. Similarly, the strain term is also used for bacterial culture, where we can use different strains of bacteria for like suppose if the bacterial strain has certain plasmid, like a different plasmid or a very special plasmid, that time we can call it as a separate strain, like that. Maybe it is of the same species, so these cells are of the same species, but the characteristics are different. So the answer is option C, cell strain. Next question. Yes, actually, here in this question, it is some like little tricky. I mean, you can see all the four options are uh, kind of will be different in neurons and glial cells. But the most appropriate option, what will be the most appropriate, if you want to distinguish a neuronal cell from like an accessory neuronal cell, what will be the most significant difference? Shape, okay, some it is to some extent different, but maybe you, if the cells are growing together, you're not able to distinguish. Size also, it might be different, but if it is like kind of some are like just dividing and some are non-dividing, so but the most significant difference is the electrical property. Very good. So the most significant difference between neurons and glial cells is the electrical property, although all other properties are somewhat different, but the most significant difference, or the difference which is like going to totally distinguish neurons from the glial cells, it is actually the electrical property. So yes, the correct option is option D. Very good. Now the last question for today. Okay, so most of you have given option C as the correct answer. But yes, Shania, yes, Parcol is actually the correct answer for cells. So cesium chloride, it's generally for smaller molecules like DNA, RNA, viruses. Because cesium chloride density based centrifugation is the density is suitable for smaller molecules. It is not very much suitable for larger molecules like cells. So cells are quite larger as compared to DNA, RNA or viruses. So for the density gradient centrifugation in case of cells, we are using Parcol, or we can use sucrose, or we can use the OptiPrep. So so the density gradient centrifugation, cesium chloride is also used, but you have to be careful for cells. For cells, actually cesium chloride is not the correct option, because as I said, it is the density like the density gradient of cesium chloride is not suitable for separating the macromolecules. Okay, so the correct option is answer A. And yes, so that's it for today. Okay, so any more questions? Okay, thank you everyone. Then we'll meet again tomorrow. Not tomorrow, sorry, next week. Okay, so bye.